Welcome back, friends. In this video tutorial we will be talking about the difficult or complex type of transposable elements which are called the composite transposons. Now what we have seen in previous case is about the IS elements or insertion sequence elements. Now for making of insertion sequence element we must have two things. One is the simple central gene region, and another thing about it, we must have two sorry extremely sorry for that. Okay, now and two regions which is flanked at both of the ends, which are called the insertion inverted repeat, sorry, not insertion, inverted repeat region, and one central gene region. Okay, so this is the actual structure of IS elements, as we can see in these previous cases. And we have also seen this IS elements are pretty small, like 780 768 BP only. So 768 BP means not 1 MB, so it's a pretty small, so less than 1 MB less than 1 MB in nature. Okay, so you can see this 768 BP in case of the galactose operon for bacterial cell. Now that pretty tell us that why these IS elements are simple, because of this length, and all these things are really simple. Only one gene at the middle and two inverted repeats at the bottom, at the ends, sorry. Now these inverted repeats are helping itself to be incorporated, and the gene portion code for the protein transposase, which is a key protein during the transposition of IS elements. Now if we look at this composite transposable elements, or suppose non-composite transposable elements, they are varying from this IS elements, because IS elements are pretty simple, but this composite and non-composite transposons are pretty complicated. Now you can see in this case, this composite or non-composite transposons, both type of transposons are made up with more than one insertion sequences, and they are not only have this more than one insertion sequence, but they can also carry a few different types of genes. Sometimes a gene which can give the bacterial cell evolutionary advantage, like the antibiotic resistance genes, right? So this kind of genes can also be taken by this composite or non-composite transposons. Now in this case we are looking at the TN10 transposons of bacteria cell. It is a total consisting of 9.3 kb nucleotide sequence, so you can see pretty big, 9.3 kb, and it includes 6.5 kb at the central region, which is the DNA, or which is the gene region, which codes for the tetracycline resistance. Not all the region, but in between this 6.5 kb we are having the gene for tetracycline resistance. So this is a very positive factor about these composite transposons, or very unique factor, that it can carry this antibiotic resistance genes, and by the means it can transfer this antibiotic resistance genes from one species to another species very few times, but sometimes, and most of the time, in between the same members of the same different members of the same species. Okay, and it also consists of two 1.4 kb sequences at both the ends, which are simply insertion sequences. We can see in this case, which are simply insertion sequences. So these two things are the IS elements, which is denoted with this red and yellow color. This yellow color portion is simply this blue color what I have drawn here, which is coding for the transposase, and these two red parts are the inverted repeats. So these two IS sequence is present at the both of the terminals of this central gene region for this composite transposons. Okay, so IS elements supply the transposase as well as ITR regions, or this IR regions recognizes the signal. Okay, so these IS elements are doing two functions. One is they are providing the transposase, which is the key enzyme for the transposition, and the second thing is this transposase, or these IS elements are also helping them to recognize ITR regions, or inverted terminal repeat, on the other target DNA site. That's also very important. Now at the middle we are having the gene. It may be different type function of genes, but in this case it is tetracycline resistance gene. Okay, so this is the structure of a composite transposon. Now if we look for the structure of a non-composite transposons, what we will find, let us move on. Yeah, this is the non-composite transposons, which is again the transposon. The example is in this case TN3, which is the type of non-composite transposons. Now again, this type of transposons can also carry genes. The example can be genetic antibiotic resistance also in this case too. So we can see here we have a beta lactamase, which is the antibiotic resistance against beta lactam antibiotics like penicillin, but in this case they do not terminate with IS elements. So that's a very important point, because in previous case, in case of composite transposons, we can see that at the central region it can carry gene, but at the both ends we are having the IS elements, because IS element must present at the both end, and this IS elements, as we know, they are consisting of the central transposase encoding region, and at the end they are inverted repeat region, and this will be flanked at the middle gene containing site. Now at the ends on IS element repeat sequences. Okay, so in this case ends are non-IS element repeat sequences. That means there must be no IS insertion sequence present at the ends. Instead of that, there must be some other type of sequence which will help them to recognize this ITR, or inverted terminal repeats. Okay, so here what we are having the left inverted repeat and the right inverted repeat in this case, and both of these repeat consisting of 38 base pairs instead of those huge 600-700 base pairs of IS elements. So what we are having a single stretch of the DNA, which DNA can really make cut onto themselves and really move from one place of the gene into another place of the same gene, or sometimes from one place of the DNA to somewhere some other DNA, right? There's the first criteria for the transposons. Second thing is that it is a pretty long element, almost like 5 kb, so it's a pretty long element. Now in between this 5 kb, only 38 BP at both of the ends, it consists of the inverted repeat instead of the insertion sequence. That's the second quality characteristic here, which is a pretty small region, and rest of all the region in typically like 4957 BP or 4.95 or 4.96 kb region is consisting of all the different types of genes. So in comparative way, we can see this non-composite transposons actually carry much more genes than the composite transposons, and non-composite transposons are not having any insertion sequence elements, and the gene sequence is not denoted, or the end of the gene sequences are not denoted by this IS elements in this case. Instead of that, they are denoted by the inverted repeat sequences at both the ends, but these are not IS element sequences. Okay, that's very important assumption. But inside that long genetic part of this transposon TN3, there must be the transposase enzyme encoding region, otherwise the transposition cannot be established. So if we find it, we can get it. So here we can see there is a region or the gene sequence present there which can code for transposons, and there are also different genes. We have TNPB, which encodes for resolvase, which is another important enzyme which help them to cut this genetic element from the DNA sequence. And at the end we are also having this beta lactamase, or some which is called the antibiotic resistance gene. Now these antibiotic resistance genes are the advantages of all, because this will help this bacteria to grow against this antibiotic environment, right? And this kind of antibiotic resistance genes transfer gives this bacterial cell the evolutionary advantage over other type of bacteria. Okay, so these are the pretty overall of non-composite transposons. So let's sum up all those about composite and non-composite transposons. You can see in this case of composite transposons, we have seen we are having the central region of the DNA that is flanked with two insertion sequence at both the ends. One is called the left insertion sequence, another is called the right insertion sequence, and at the middle it may carry gene. For example, it can carry the antibiotic resistance gene. Here in TN10 type of transposon, it can carry the tetracycline resistance gene. Okay, and this type of transposons are pretty long, like 9.3 kb at its size. And at the second hand, we are having the non-composite transposons. In case of non-composite transposons, we are having a long stretch of DNA at the middle, and pretty fewer stretch of nucleotide sequence at both the ends, but still they are not insertion sequences. Instead, they are only inverted repeats, left and right inverted repeats respectively, and at the middle region where they have different genes, they can carry more than one genes. Here for example they carry 3 different genes. One is the gene for coding transposase, another gene is coding for resolvase, and the third one is coding for the unique advantage of the bacteria, which is the beta lactamase. Okay, so this is all about the composite and non-composite transposons, and I hope it will help you. Thank you.